We're now going to look at examples of the thyroid and parathyroid, starting with slide 13b, which is a plastic section of a normally active thyroid. At low magnification, we can see that this is a lobulated structure. At higher magnification, we can make out individual follicles, which are the functional units in the thyroid. At higher magnification, we can see the individual follicles lined by their simple cuboidal epithelium. The pink contents is colloid and is secreted by the follicular cells into this lumen. At higher magnification, we can see the cuboidal thyroid follicular cells going all the way around, as well as another cell type called the C cell. Here are a couple of C cells which secrete calcitonin. They are not in contact with the colloid but are separated by the follicular cells. You can also make out the capillaries in between individual follicles. The next slide is 14b, which is a paraffin section of a thyroid. It has a portion of the parathyroid as well. So here we can see the parathyroid, which looks quite different than the thyroid. You can see the individual follicle. One difference between a paraffin section and a plastic section is that the colloid is partially extracted, so you get these white areas or white circles within the colloid. You can see the cuboidal epithelium of the thyroid follicular cells around the follicle, the colloid in the center, and the white spots indicating some partial extraction. It is more difficult to identify the C cells in the paraffin section. At higher magnification, we can compare the thyroid follicular cells to the chief cells in the parathyroid, which are densely packed and have very little separation between them except for by blood vessels. The next slide is 16b, which is a highly active thyroid. As we go up in magnification, we can see that the colloid volume is lower than in a normally active thyroid, because the thyroid follicular cells metabolize the colloid material to produce active hormone much more rapidly. So here is colloid material, here is the wall, which we can see is now not a regular oval but invaginated in places. We can see the red blood cells associated with the capillaries surrounding these follicles. We're now going to look at a 22b, which is a paraffin section of the parathyroid. At the intermediate magnification, we can see that this is a highly lobulated structure. Connective tissue is surrounding the individual lobule. At higher magnification, we can see that the lobules contain two cell types, the darker or more basophilic chief cells, and then in another cell which stains more eosinophilic, which is the oxyphil. So chief cells and oxyphils. Oxyphils represent inactive cells that transition from active chief cell to oxyphils, which contain many mitochondria, giving rise to this highly eosinophilic appearance. So this lobule contains a mixture of active chief cells and inactive oxyphils. We can see here another lobule which has almost all inactive oxyphil. There's one area down here where there may be some active cells. So some of these lobules have transitioned to inactive forms and others are mostly active.